Dr. Kim, you can start sharing your slide. So Dr. Kim is an assistant professor in also Department of Microbiology and Immunology and from Seoul National University College of Medicine. And his title is Simultaneous Epigenetic and Gene Expression Profiling of PBMC from COVID-19 vaccination patients, focusing on Treg cells as well. So over to you, Dr. Kim.
Hey, thanks for the kind introduction. Everybody are hearing me?
Yes, it's good. And slides.
Yeah, okay. I think that I need to focus on the regular T cells following our mentor's talks. So today I'm going to talk about the topic, the simultaneous epigenetic and gene expression profile of PBMC from COVID-19 vaccination patient focusing on Tregs. Actually, I'm not a very big guy like my mentor, so I want to introduce myself first to your group. Actually, I was born in Tongyeong. We call there as a Naples of the Korea. It's very, very beautiful city. If you guys have a chance to visit Korea, do not please visit our hometown also. Everybody wanted to visit the Seoul and the Jeju Island, but Tongyeong is also a very, very beautiful city. And then let me summarize my career first. Actually, I got an MD in Seoul National University College of Medicine, and I became a dermatologist under the Samsung Medical Center. And then I first met immunology when I became a student of the Professor Park in 2012. And from 2012 to 2017, I did a lot of the in vitro work and in vivo work, especially focused on the transplantation immunology and also the xenotransplantation under Cheongju Park. At that time, I learned about how to work, and then I learned about regulated T cells to control the autoimmune disease and transplantation rejections. And after that, I moved to the US and I got a postdoc training under Emma Gatmiyaski in the New York. And there I learned about the high resolution omics tools and came back to South Korea. This is a summary of my the training course. And also I really love to play the soccer. And so if you have a chance to have the game with the if there is a kind of the soccer team in the University of Malaysia, then hopefully we can have a soccer camp with your institute. And when I was a graduate student, I published several papers on the Cheongju Park, focusing on the xenotransplantation, second the regular T cell, and also we had domain analysis on CD40 and CD40 ligand costimulatory blockade cosmulation part. So I published several papers, but in fact that was a hard time because at the time I did the in vitro, in vivo kind of the hardcore work, but always we could not get a very, very good kind of expected result. So I found the research is very difficult. But what I got an insight, the insight what I got when I was on the Cheongju Park is that regular T cell is very important. So to control the autoimmune disease, we human bodies developed the cell subset called as a regular T cells, and then we had this very, very big xenotransplantation project. So we depleted all T cells in monkey and transplant the porcine islets, pig islet to the monkey, and one group was transfused with the regular ex vivo expanded regular T cell, and one group did not have the Treg infusion. What we found is that if we transplant actually the cell transplant the regular T cell, ex vivo expanded regular T cells in the T cell depleted the animal, we found that the porcine island, xeno island, is being survived longer than the other groups. So we published this work in HAT, one of the paper in transplantation research, but have not dissected the mechanism how the Tregs can work in these organisms. So still, this is the longest record we have in this world. So more than 1,000 days that the porcine islets were survived in the monkeys. So after I got an insight that the regular T cells is very important in a lot of field, so I moved to the US and did some targeted single-cell proteomics technologies called the Cyto. The team was like that way, and I've collected 15 different healthy control blood together with the atopic dermatitis patient blood. And the one thing I want to emphasize on is that always sex age matched control is very, very important. And in where I'm working in the US, we have to think about the ethnicities, but the most important thing while we are for this kind of work is that the sex age matched control. So I've collected 20 atopic dermatitis patient blood, and then collected another 15 sex age matched healthy control blood, and did the Cyto like that. So the Cyto is kind of the cutting edge technology tools that can profile the surface and intercellular proteins at a single cell levels. And we designed this kind of panel. So it means that we can profile all of these markers expression levels at a single cell levels from the PBMC or the tissue dissociated single cells. And also we did we stimulated the immune cells by PMA ionization, and then have checked this kind of the cytokines that the cells can secrete after the stimulation, and we established this kind of the pipelines, that the analytic pipelines, and found that this is the whole Tregs. The right side is the atopic dermatitis. This right side is the regulatory T cells in atopic dermatitis, and left side is the regulatory T cells from the healthy control. We have done this study with the circulating immune cells, PBMC, and very reasonably we were surprised as that on the right side in atopic dermatitis, that only the CLA, cutaneous leukocyte associated antigen having regular T cell, so frequency is being increased in the circulations. The CLA positive Tregs is just compromises 5 to 10% of the total circulating Tregs, and very reasonably in the skin disease having the atopic dermatitis patient blood have increased frequencies of the CLA positive Tregs. It means that to control the immune response in the skin, a lot of Tregs are migrating into the skin and proliferated, and is being migrated out to the circulation. We are observing the kind of the phenomenon, and after that I dissected these cell populations and found CD38, this is one of the functional markers. CD38 is being decreased in the CLA positive Tregs in atopic dermatitis blood. And then what I found is that CD38 can be interpreted as important functional markers on Tregs. So if the Treg have a lot of CD38, the Treg is strong. And if the Treg have a reduced level of CD38, those function will be decreased. So reasonably, what I think is that the CLA positive, the regulatory T cells in CLA positive compartment in atopic dermatitis patient blood, the function will be impaired. So that would be another cause of the kind of the inflammation, because the regular T cell can control the inflammations. So to extend this kind of to extend this research in South Korea, and I with the Professor Park, and then we did the extension study. And as you know, in this field, regular T cell field, there is 3 giants named Simon Sakaguchi, who first found the regular T cells, and Jeffrey Fluson, actually my mentor's mentor. He recently found the Sonoma Biotherapeutics after he completed his academic career in UCSF, and he even though he's a PhD, he's more focus on treating the disease by using the human regulatory T cells, and then finally the Alexander Brzezinski, who had a very fancy mice models. And then recently the Simon psychology are focusing on the epigenetics of the regulatory T cells. We searched his recent papers and found, and then he said that ETS1 and ROS1 and STEP5, another kind of the transcription factors that will be important in maintaining the function or enhancing the function of regular T cells were reported in 2020 immunities, and then we decided to check these kind of molecules, including FOX3, in our data set, and after we got a government fund, we collected patient and human blood and sorted enriched CLA positive Tregs in each patient. And even though it's kind of just one slide, it took more than 18 months to complete this project, and found that in atopic dermatitis patient blood, CLA positive regular T cells having less expression levels of ETH1 and then IKGF2, the Simon psychologist said, the molecule, the Simon psychologist said that these are the these are very important transcription factors in regular T cells to maintain the function. So, but this is the PBMC work, so systemic work. So every reviewers might be asking about us, well, what's going on in target tissue. So we downloaded the single-cell data from the public. So everybody can download the single-cell RNA-seq data in the publications. And actually, this work have done, this experiment have done by me when I was in a postdoc in the US. And these are 20 patient, the atopic dermatitis patient skin single-cell RNA-seq data, and we downloaded the data and we processed the data. We found that more than 500 T cells, more than thousands of the T cells were found in the atopic dermatitis skin single-cell RNA-seq data, and annotated regular T cell, and found ETS1 and ROS1 and STEP5 are indeed being decreased in atopic dermatitis patient skin. So we are doing the functional studies on this research, and that now we are scaling up this research. So we have collected with a collaboration with domestic dermatologists who are interested in this work, and we've collected more than 200, currently more than 50, the patient blood, especially from the severe atopic dermatitis patient, and found then we found that these results were recapitulated. So, and following the disease severity, so it means that the healthy compared to healthy control, the severe atopic dermatitis patient blood have the increased frequency of the CLA positive Tregs, and also the Th2 Treg that is already known. So we are analyzing this data now. That this is, as far as I know, the largest single-cell RNA-seq data in the world regarding atopic dermatitis. And as I told you yesterday, and we have very big consortiums. More than 50 PIs were involved in this project. We call the project as a SCADE, Single Cell Atlas of Inflammatory Disease. So we are profiling more than 16 different immunologic disease across the domestic. After we collect the samples across the big hospital in South Korea, the PI is the Jong-il Kim, and then in the immunology part, the Chung Park is had a leadership on this project. So I think that is the introduction part. If you think about the titles of my talk, so the question I have is that whether regular T cells in acute infectious condition is being exhausted. So because what I found is that the regular T cells in the chronic inflammatory conditions like atopic dermatitis is being exhausted or dysfunctional because of, I think, that the clonal expansion of those cells. So I really wanted to see what's going on systemic regular T cells in acute infection conditions like the COVID or acute viral infections. So I really wanted to profile the patient blood who experienced acute infection or the vaccination. So this is first one is my personal motivation to do this kind of project, but government asked me to decipher the differences between the systemic immune response between the adenoviral vector associated vaccine like the AstraZeneca versus lipid nanoparticle mRNA vaccine like the carrier-free vaccines like the Moderna and Pfizer. So this is the 1 year project because the COVID project have to go fast, and then we strategically collect the patient blood here and there, and then we collected actually 16 different 16 the carrier-free mRNA vaccinated patient blood and the adenoviral mRNA vaccinated blood. And also in South Korea, we started to vaccinate the protein vaccine, the conventional vaccine who don't really want to get the mRNA vaccine shot. So even though they are very few, we successfully collected patient blood from 12 different protein vaccinated patients and also healthy control. And then we after we got a PBMC from those patients, we have to profile the epigenetic signatures together with the mRNA signature. We have to isolate the nucleus of the PBMC. So the sample should be very good because while the isolating nucleus in a single cell level is not a very easy work. So the QC criteria for this kind of work is more higher than a conventional single cell RNA work. So we successfully got a patient blood who have a higher viability and then our QC criteria, and then the sufficiency and the numbers that we have, the sufficiency and numbers in each file, and all of this patient information is collected in the central, and then we are now analyzing the data, and I think that this data will be coming out early next week, and then compared to the healthy control, the COVID vaccinated, the protein vaccinated patient and the cross vaccinated patient have some kind of the differences of frequencies in certain set immune cells like the memory B cells and the CD4 central memory cells and then effector cells. But these are the preliminary data, so only the 8 versus 8 patients. So we are adding the data now, and actually I really wanted to introduce the spatial transport platforms while for the pathologists who are listening to our talk, but I think that because of short time, I will talk this part later. So I think that this is the right talk, and if you have any questions, please feel free to contact us. Thank you.
Right, thank you. I think we do have some time for questions. So anyone have questions, you can type at the chat box. At the moment, okay, maybe I will ask, since Dr. Kim is here, I will ask Dr. Kim first. From Kamala Kanan, basically the question is, can the vaccine for COVID increase the T cells?
Increase the T cells?
Yeah. Can the COVID vaccine
actually
increase the T cells?
So that's quite important questions. There's a lot of applications by using this kind of multiomics tools, and the answer is no, actually. So we haven't observed that the frequency of the T cells in the circulation is not being increased after the COVID vaccinations. But we found is that certain type of the T cells, like the COVID specific T cells, are being definitely increased after vaccinations.
Okay, great. Okay, over to you, Professor Park.
Yeah.
Kamala also asked another question.
Fasting increase the T cells?
Yeah. Can fasting increase the T cells, and can we live without T cells?
So I don't think the fasting increase the T cell. T cell, actually immune systems, the key maybe characteristics would be the balance. So the T cell also depend on the nutrition. So I think fasting may, decrease the function of T cells, but I'm not sure whether the number wise it could be, changed by some exogenous stimulus or environment, because the homeostasis of the immune system is very robust, and we could not live without T cells. T cells is very critical in protecting ourselves from the environmental invaders such as bacteria and virus. So for example, the AIDS patient died by the infection because of they have the T cell deficiency.
Okay, so that explained very well. And Professor Sakunan asked, can regulatory T cells can be made available commercially for treatment of rheumatological disease?
Yeah, currently it's in testing in clinical trial. It's not been commercialized yet. So if we have a safe and efficient data from the clinical trial, then it could be commercialized. It's not yet.
Okay. So for everyone's information, Professor Park is actually the Chief Executive Officer of PB Immune Therapeutics. So that's actually his product PB201 or 301, right? Okay. So Professor Park, maybe I have a question for you. So at the moment, your drug, it's currently in which phase of clinical trial?
So it's in the preclinical stage. So we are now preparing a cell line which meet the regulatory requirement. Then we could move on to the phase 1 clinical trial, whether it is safe or not. And then we could move to phase 2 or 3, so that we could compare the effect of regulatory T cells with the current treatment modality, whether it is better than the current modality. Then, it could be commercialized.
Okay, so do you see that COVID-19 we know COVID-19 vaccine can be marketed in a year, right? So will this revolutionize the way how we actually also do clinical trial for products like all this monoclonal antibody for immune diseases?
Yeah, actually, currently, many monoclonal antibodies is in the market through the clinical trials. So we have so many experiences in testing monoclonal antibody as therapeutics by clinical trial. So, and also it is true, it is true with the cell therapy. So yeah, COVID-19 delayed the clinical trial because we have very difficulties in recruiting patients because of the COVID, but now it became better and better condition, so that a lot of clinical trials is now going on.
Okay, I think in lieu of time, we have to stop here because
we have to
catch a flight.
The taxi is waiting.
Okay, so for anyone who has more questions, are interested in their work, I can share the email, and please contact Professor Park as well as Dr. Kim, and they're happy to recruit talented Malaysians to join their lab and their team. So thank you so much. It's really a great honor to have you with us despite your busy schedule. Thank you so much, and thank you for those who attend. And as mentioned, please register to claim your MMS-CPT points. Yep. Okay. Thank you everyone for attending. Thank you, Prof. Park and Dr. Kim. Safe journey to Bangkok.
Okay. Thank you for having us. Yeah. Bye.
Bye. 감사합니다.